Imagine you run every toxicology test, every stability studies, every analytical method, and your drug product passes. Months later, a regulatory agency flags a compound in your drug product that was never on your radar. Not from your API, not from your excipients, from your rubber stopper. That's an extractables and leachables. And for thousands of patients in the early 2000s, the consequences were life-threatening anemia. Hello, I'm Raghuveer Singh Parmar, Director Scientific Affairs at Resolve Mass Laboratories, and I work with pharma and biotech teams every day to design and execute ENL programs that protect patients and move submissions forward. Today we are covering the essentials, what extractables and leachables are, where they hide, why they matter, and what a solid ENL workflow looks like. For most of pharmaceutical history, the container was just the container. The idea that a rubber closure or a polymer bag could pose a patient safety risk wasn't part of the regulatory conversation. Then in 1998, a reformulated version of epoetin alpha Aprex changed that permanently. A new excipient, polysorbate 80, replaced human serum albumin as a stabilizer. That combination interacting with a leachable from the rubber stopper triggered a cascade of immune responses. By 2003, over 13,000 cases of pure red cell aplasia has been reported globally. The industry learned at enormous cost that the container is part of the drug product. So what exactly is the difference between extractables and leachables? These terms are often used interchangeably, but they describe two different things. Extractables are compounds released from packaging or device components under exaggerated laboratory conditions. Aggressive solvents, elevated temperature, extended contact times. They represent worst case potential. Leachables are what actually migrates into your drug product under normal, real-world conditions of storage and use. Every leachable traces back to an extractable, but not every extractable becomes a leachable. That distinction drives your entire testing strategy. The extractables profile tells you what to look for, and leachable study confirms what's actually there. So where do these compounds come from? Where do leachables actually originate? Any material in direct or indirect contact with your drug product is a potential source. The highest risk category is the container closure system. Rubber stoppers, elastomeric plungers, polymeric bags, plastic vials, and the caps and coatings that seal them. But something most overviews on this topic skip manufacturing contact materials, tubing, gaskets, pump heads, filter housings. These aren't packaging, but they touch your drug during processing, and they are fully in scope under ICH Q3E. The scope is wider than most teams expect when they first map it out. Why regulators take this so seriously? Why does regulatory scrutiny on packaging materials run this deep? Three interconnected reasons. First, patient safety. Injectables and inhalation drugs deliver leachables directly into systemic circulation. There is no first-pass metabolism to buffer exposure. Toxic, genotoxic, or immunogenic compounds at microgram levels can cause documented harm. Second, regulatory compliance. Missing or inadequate ENL data is a consistent source of FDA complete response letters and EMA deficiency notices. ICHQ3E, finalized in 2024, has unified the global bar. There is no safe harbor for underdocumented submissions. And third, product integrity. Leachables can degrade your API, shift pH, create visible particulate, and accelerate degradation across a product's shelf life. Not all dosage forms carry the same risk. Risk is not equal across the dosage forms, and the depth of your ENL program should reflect that. At the top of the risk ladder sit parenteral and inhalation products. These have the most direct route to patient exposure, and FDA's expectations here are well established and uncompromising. Ophthalmic, transdermal, and oral mucosal products sit in the middle tier. Direct contact with sensitive surfaces demands a systematic approach. Oral solid dosage forms carry lower inherent risk, but that doesn't mean zero. Specialized packaging like unit dose foils, moisture barrier films, and blister push through systems still need to be assessed. ICHQ3E applies a risk-based framework precisely because one size does not fit all. When ENL goes wrong, real consequences. The stakes here are not theoretical. At Resolve Mass, we have supported sponsors through ENL investigations that started with a single unexpected analytical result and ended in full reformulations, closure replacements, and refiled submissions. Each one expensive and in retrospect avoidable. The Aprex case remains the most cited example for good reason. An invisible compound at trace levels interacting with a newly introduced excipients caused life-threatening immune response in over 30,000 patients. The manufacturer had to reformulate, replace all closures globally, and rebuild the safety dossier from scratch. That pattern, invisible molecule, catastrophic consequence, retrospective remediation appears across the industry. The cases look different on the surface, the lesson underneath is the same. So 5-step ENL workflow. A well-designed ENL program follows 5 steps, and the order matters. First, identify every component in your container closure system and manufacturing contact surfaces. Second, run control extraction studies under exaggerated conditions, selecting solvents and temperatures that represent realistic worst-case scenarios for your specific formulation. And third, analyze those extracts using GCMS, LCMS, ICP-MS, and headspace methods to build a complete extractable profiles. Fourth, assess each identified compound against ICHQ3E thresholds, the analytical evaluation threshold, the safety concern threshold, and qualification threshold. Fifth, document everything for your CTD Module 3 submission package. The teams that build this in early save themselves months of pain. The ones who bolt it on at submission almost always find something they didn't expect. So what should you walk away with this today's discussion? Extractables and leachables are two distinct things. One is potential, one is real, and conflating them leads to poorly designed studies. ICHQ3E gives you the global threshold framework. Know your AET, your safety concern threshold, and your qualification threshold before you design your study. patient harm from leachable is documented. The Apex case closes the door on treating ENL as a formality, and early assessment always beats late remediation. Next up, we go deeper how to design the extractable studies, choosing the right extraction conditions, solvents, and analytical battery for your specimen container closure system. Don't miss that future videos in the series. If you are working on an IND, an ANDA, or a biologics program and you need a CRO partner who treats extractables and leachables more than a tick box exercise, Resolve Mass Laboratories supports a small and mid-sized pharma teams from first material assessment all the way through submission. Thank you so much for watching this video, and see you in the next video. Until then, goodbye for now.